So today when we start with epistasis, if you remember this picture where I had showed you the difference between dominance and incomplete dominance, and I had said that if the heterozygote looks shows features, morphological features very similar to the dominant allele, then we consider that the dominant allele is capable of totally masking the effect of the recessive allele. Since the dominant allele is capable of totally masking the effect of recessive allele, as you can see from this picture, epistasis is something very similar to this, but there is a point of difference. If you look, A1 is one allele, A2 is the other allele. If A1 is totally dominant over A2, then in the same locus, A1 is able to totally mask the effect of the recessive allele. Now I said epistasis is very similar to dominance, but the two alleles in concern do not belong to the same locus. Here A1 and A2 belong to the same locus. But when we think of epistasis, epistasis is nothing but a very similar kind of interaction where one allele of a gene is totally masking the effect of another allele of a gene. So there is more than one gene brought into consideration. So I started with dominant to make you understand that in dominance we have studied that one gene masks the effect of another gene. Sorry, one allele masks the effect of another allele. But here, in case of epistasis, here also one allele masks the effect of another allele, but the two alleles in question do not belong to the same locus or same allelic group. Okay. So with that, we start our epistasis. Where is the right? Now let us look into this picture. This is a let us look into the parental cross. Capital B, capital B, capital E, capital E. Now let me first say that this is a story of the Labrador dogs, and capital B allele codes for the black color of the coat. The small b allele produces the brown pigment. So the capital B allele produces the black or the dark pigment, and the small b allele produces the brown pigment. So this is the story of these two alleles. So there is a capital B allele and there is a small b allele. Again, capital B codes for black pigment, small b codes for brown pigment. Now let us look at this allele, capital E. Capital E is the extension locus. Capital B is the black locus. Capital E is the extension locus. Another point, for capital B, black is dominant over brown. Now let's look at the extension locus. The allele of this locus, what it does, it deposits the pigments on the hair shaft. So dogs have hairs. So if it's a brown pigment or a black pigment, the E allele, capital E allele, deposits the pigment on the hair shaft. Okay. So if it's the capital E allele is present and we have a capital B allele, B codes for black pigment. E allele now E allele now deposits this black pigment on the hair shaft, and thus the hair color becomes black. But this E allele, this is the recessive of the extension capital E allele. This E allele is unable to deposit any pigment on the hair shaft. So if no pigment is deposited on the hair shaft, then by default the color of the hair becomes yellow. So when we have a capital B and a capital E combination, we get the color black because capital B produces black pigment, capital E can deposit this black pigment on the hair shaft. When we have a small b capital E combination, small b produces brown pigment and capital E is able to deposit the brown pigment on the hair shaft. But now look at the other combination. When there is this homozygous recessive condition, as you can see, E is written here, prevents the deposition of dark pigment. So when there is small e present at this locus, E will prevent the deposition of either the black pigment or the brown pigment, as a result of which the color of the hair becomes yellow. So the presence of these two homozygous recessive allele actually masks the effect of small b or capital B or capital B small b combination. In other words, the presence of small e in homozygous condition in one locus is preventing the phenotypic expression of a gene or an allele set in another locus. So small e is actually preventing the expression of the capital B small b phenotype. So when we have a parental cross like this and we have a F1 heterozygotes. Now the F1 heterozygotes are self-fertilized. So when they are self-fertilized, if you remember the Mendelian dihybrid cross, what are the genotypes we get? We get a black genotype with, since capital B is completely dominant to small b and capital E completely dominant to small b. So whatever we write here becomes immaterial. It can be a homozygous capital B or a heterozygous capital B small b. The phenotype will be of that of capital B. Similarly, in this dash, whatever we write, we can write a capital E or a small e. Whatever we write, this capital E expression will sustain. So the phenotype will be black because we are having this E allele which is capable of depositing the dominant pigment, which is black, on the hair shafts. When we have this combination where we have a homozygous BB allele producing the brown pigment and a capital E, one dose of capital E or a double dose, this capital E is able to deposit the brown pigment on the hair shaft and we get brown hair color. But when we have small e, as I said, small e is unable to deposit any kind of pigment on the hair shaft. So if there is a capital B or there is a small b, it's irrelevant. Small e will not deposit either the black pigment or the brown pigment, as a result of which we get these two categories gives us yellow colors. So you can see that is the deviation from the original ratio of 9:3:3:1 to 9:3:4. So this is a case of recessive epistasis. Again, I am saying why epistasis? Because a set of gene is masking the effect of another gene. Here, if you see in this particular example, the small e is able to mask the effect of the other set of alleles in the other gene. So the gene that is masking, so small is a recessive set of gene, it's a homozygous gene. So the gene that is masking is known as the epistatic gene, in this case small e, and the gene that is being masked, whose phenotypic expression is being masked, is the hypostatic gene, in this case capital B, small b. So these genes are being masked and this is masking. So as you can see, this gene is masking, so this is the epistatic gene, and this gene can only mask the effect of the other allelic phenotypes in its recessive condition only, hence recessive epistasis. So this was about recessive epistasis. So just for once more, that the gene which masks is the epistatic gene and the gene which is being masked is the hypostatic gene. Okay. Now coming to another example. So this is a recessive epistasis. So I'll be going for another example of recessive epistasis, specifically with that blood group 1. Because I had said you last day while teaching that we'll come back to this Bombay phenotype concept again, because there is an epistatic interaction going on. So we should always remember that when we think of epistasis, it should be a dihybrid situation and there should be two genes in consideration. Okay. Now, if we recapitulate what we did in the last class, I'm going very slowly. This is the H compound. This is the intermediate H and we have the H compound. And if we remember the blood group, there were two alleles, IA, IB, and there was a IO condition. So blood group fell under multiple allele concept. When we have a IA, now if you remember what we did in last class, is I'm again recapitulating, IA is codominant to IB, IA is completely dominant to IO, IB is also completely dominant to IO, but IA and IB are codominant. So having this basic concept, so when we think of IA blood group, so there is a IA allele, it can be present in a homozygous condition or in a heterozygous condition to IO. Mind it, the combination will be IA or IA-IO. In such a condition, if you remember, what does this IA antigen? It used to code for IA allele codes for the enzyme glycosyl transferase. And this enzyme simply adds an N-acetyl galactosamine to the H compound, as a result of which we get A antigen. We did it last class. Again, we have the IB type. In the IB type, what can be the combinations? IB, or IB-IO. Both these combinations will give us B blood group. Here also, the IB allele codes for a glycosyl transferase that adds galactose to the H compound. So, galactose is added to the H compound. Okay. So we have a IA locus and a IB locus, and as you can see, the IA allele adds N-acetyl galactosamine, we get A antigen. The IB allele adds galactose, we get the B antigen. But in case where there is neither IA or IB, but there is IO-IO, as you can see, there is IO-IO, so we have the H compound. No terminal sugar is added. Now comes the peculiarity of the case, which is known as the Bombay phenotype. Okay, let's look here. Suppose you can see this is the H compound, the H locus. There is a mutation and we get this combination, small h, small h. So this is a mutant. Okay. Since this is a mutant, no H compound will be produced. And if so, we have only an intermediate precursor. No H compound produced. And if no H compound is produced, then we cannot have A, and even if there is a IA allele, since there is no H compound here, IA allele will not be able to add A antigen, or IB allele will also not be able to add the galactose terminal sugar. So, when there is a HH, there is no H compound. So, even if there is a IA allele, no addition of an acetyl galactosamine. Even if there is an addition of IB, even if there is a presence of IB allele, no addition of galactose to the H compound. As a result of which, what we get in the blood of those people, we get this one, which is a similarity to the character of the O blood group, because you see neither the terminal galactose nor the terminal galactosamine has been added. So this is a picture that we observe in the O blood group. Even if they may have a presence of a IA allele or a IB allele. So again in this case, the homozygous recessive condition of this HH at one gene is actually masking the phenotypic expression of IA allele or IB allele in the ABO blood group locus. So this is a case of, again, another case of recessive epistasis. So if we look at the genotypes I've shown here, when we get a A blood group, the combination is H substance C. This is the H compound and the IAIA, H compound and the IAIO. H compound can be present in the homozygous or the heterozygous form. Doesn't matter because one H is sufficient. These are haplosufficient genes. Again, for the B blood group, you see we have a H compound. The blood group allele is IBIB or IBIO. For the AB blood group, we have the H compound and the blood group allele is IA and IB. As it is codominant, we have AB blood group. Now you see, we have a H antigen, this H compound, and II, no addition of terminal sugars, O blood group. Now we have mutant. So whenever we have HH, whoever sits at the blood group locus becomes immaterial because all the genotypes will exhibit the phenotypic expression as well as the genotypic characteristics of the O blood group. So the HH allele is totally masking the IA expression or the IB expression. So this is another beautiful example of recessive epistasis. Okay. Now finally, so this is where we saw that a recessive gene, whenever a recessive gene is actually masking the effect, you have to keep in mind that the recessive gene is present in a homozygous condition, because in a homozygous condition only the recessive genes are capable of expressing their effects. So you have to keep that in mind, that whenever there is a masking of the recessive gene, recessive epistasis, then we have to consider that the homozygous recessive gene is actually, the homozygous recessive condition is actually masking the effect of the other gene. Now we come to dominant epistasis. This is a example of dominant epistasis. Before going into the cross, first let me show you the pathway. So there are two alleles in this cross also, as you can see. There is a W allele and there is a Y allele. And as you can see in this particular cross, which I have shown here, in case of the W allele, what are the functions? Let us look by this pathway. Let's know the pathway first, then I'll go to this cross. These are squash fruits, okay. Squash is a vegetable, not a food. Anyway, these are squash, and the W allele encodes for an enzyme or helps in the synthesis of enzyme. Let me tell you this pathway first. This is a white fruit, white squash, and the color of this white squash is due to a compound A. Let us imagine. The compound A is converted to compound B by enzyme 1, and the compound B is converted. So the compound B gives a green color to the squash, and the compound B is converted to compound C by enzyme 2. So this is a pathway by which color is generated. So compound A is converted to compound B, which gives a green color, and compound B is converted to compound C, which gives the yellow color. The conversion of compound A to compound B is brought about enzyme 1, and the conversion of compound B to compound C is brought about by enzyme 2. So when the pathways are normally functioning, we get the yellow color. If there is somewhere disturbances in the pathways, we get the intermediate colors. Now let's look at the allele. As I said, there is a W allele and there is a yellow allele. Now, what does the W allele do? As you can see, it represents the dominant allele. W is a dominant allele that inhibits pigment production. So no pigment is formed, is albinotype that inhibits pigment production and produces white squash. So as you can see, when there is a capital W allele in one dose or two doses in a homozygous or heterozygous condition, it prevents the function of enzyme 1. So if enzyme 1 function is prevented, then automatically the fruits, the squash will stay white in color. So the W allele is actually preventing the synthesis of enzyme 1, as a result of which they prevent the pigment formation, so they do not produce this enzyme 1. It is not produced by them. So if there is a W allele and if there is no enzyme 1 produced, this pathway stops here itself. It cannot move further. So all the plants having one dose of W allele or two doses of W allele will appear white due to the absence of enzyme 1, as you can see here. So you have a one dose of W allele or a two dose of W allele, doesn't matter. When we have a capital W, we have to imagine that the squash color will be white. Okay. So W is in fact not even bothered to see what Y is actually producing. So the dominant allele here in a single dose, that is in a homozygous condition or in a heterozygous condition, can completely mask the effect of the capital Y allele. Now let us see that if the plants have a WW allele, the WW allele produces the enzyme 1, which converts the colorless compound A to the colored compound B, which is green. So plants with WW can produce green squash. But we have another allele also, that is a Y allele. Now plants with capital Y produce enzyme 2. And the enzyme 2 is able to convert compound B to compound C, that is yellow color. So when we have a W condition and one capital Y or two capital Y, we will be getting the full pathway. So those plants will have yellow squash. You can see W and one capital Y. We get yellow squash. The full pathway is operative. But we have W plants, small WW plants producing enzyme 1, and we get the green color. Now we have small YY. Now small YY genotype also do not code for the enzyme 2. So in that case, so WW plants, enzyme 1 produced green squash, and the YY plants cannot produce the enzyme 2. So the plants remain in their intermediate stage, that is the green squash. So you can see if we have a combination of WW and YY, we get green squash. So this is also a variation from the Mendelian dihybrid ratio of 9:3:3:1, where we are observing a dominant epistasis 12. This is added up because capital W is masking the effect of capital Y and small y. Whoever is sitting in the other locus doesn't matter. Capital W is able to mask the effect of both of them. So